Good afternoon once again, and welcome back to SCH 214, Intermediary Metabolism 2 and Biomembranes. I understand that after today's lecture, we'll be having 2 more lectures, and then you'll have 1 week before you start exams. That is to the best of my knowledge, you are going to start exams 18th of July. That's what I know. So if you don't know about it, I'm telling you. So if you have not started actively preparing yourselves towards exams, I will urge you. You still have some time. Start doing so. Little drops of water make a mighty ocean. So you don't wait till the last minute before you want to cramp everything into your head. It will be disaster. So little by little, I sent you self-assessment questions. You have the presentation slides. And I mean, if you go to the library or even you are on the internet, you use any biochemistry source and use the presentation slides as a guide, you can study well and prepare for your exams. Hopefully next week, we'll be able to meet face to face. Only that I don't know if it has to be, I mean, the time, because the original time that we're supposed to meet, we had changed it because of the situation where I have to be teaching you online. So, class rep, you should let me know over the course of, I mean, after today, maybe by over the weekend, you should send me a message, let me know when we can have our lecture next week, and give me details of maybe the auditorium where we are going to have it and all of that, because I'll be in Ghana, so we can meet face to face. If we can meet at least the next 2 lectures that we have left, we meet face to face before you start your exams, that will be fine. All right, so today we're continuing with the biomembranes part. We are done with the enzymes. The last time we did look at some industrial applications of enzymes. We ended the chapter of enzymes there. And so, that is intermediary metabolism. The intermediary metabolism part, we ended it there. So we're continuing to, take a look at biomembranes. And today we want to look at composition of biomembranes. What are biomembranes composed of? And I've decided to make your lecture in such a way that we don't pay so much emphasis on the structure of the lipids, because we did a lot of lipid structure last semester when we were doing the intermediary metabolism, and we came to lipid metabolism. We did a lot on lipid structure, and we looked at different types of lipids in membranes, and numbering of carbons. We did all those fundamental things on lipids. So we'll just go straight to talking about, getting to understand the membrane structure with the lipids in it. And then we continue from there and look at other components that we find in the biomembranes. So it's a build-up on what you already know, so that we don't have to repeat some of those things which you did last semester. So first of all, we want to understand what biomembranes are. These are selectively permeable barriers that separate the interior of a cell. If you are talking about a cell membrane, then it separates the cell from its external environment. But if you are talking about an organelle, which is inside the cell, then the biomembrane separates the organelle from its environment, and that is specifically the cytosol. I mean, the liquid part of the cytoplasm. So the cytoplasm is made up of that cytosol, which is the liquid part, and then you have these organelles that are swimming in the cytosol. And so you have biomembranes, sort of creating an intracellular, or you can say an intraorganelle compartment, separated from its external environment. And we say it is selectively permeable because it allows some substances to pass through it, but not all of them. And we'll come to see some mechanisms of transport, how substances go across the biomembrane in and out of cells or in and out of organelles, but particularly we just focus on in and out of cell membranes and not so much on the, I don't think we'll take a look at any example of, say, organelle, except that I think even in when we were doing the intermediary metabolism last semester, we did even look at some aspect of transport across mitochondrial membrane. If you remember, you can go back and look at that, where we did talk about metabolism of lipids. You can go and see there's some specific aspect of transport of lipids across the mitochondrial membrane. But in this case, we're just going to focus on transport across the cell membrane. And as you can see, you have a simple structure of an eukaryotic cell, and that eukaryotic cell has a cell membrane around it, and inside of it you have nucleus, and you see the nucleus is also having a nuclear membrane. And you have the mitochondrion, it's also having a mitochondrial membrane. So you have the cell membrane, which creates a barrier for the cell across, separating it from its environment, and inside of it you have those organelles there. Even if you take a prokaryotic cell where you don't have a distinct nucleus, where you have the nuclear material sort of spread in the cytoplasm, you still have the cell membrane before you have a cell wall for such a prokaryote. Right. So having understood that what the cell membrane, these biomembranes are, the composition of biomembranes is mostly the phospholipids. So you see, we studied phospholipids, the structure of it. So you can go back and look at how phospholipids look like, because we will not be going back to that. And these phospholipids form lipid bilayer. And within this lipid bilayer, you have some proteins that are embedded in it. And you have some glycolipids. That is a complex of carbohydrates and lipids. And so you see, we did study the structure of carbohydrates, we did study the structure of lipids. And so you just know that these structures can form a complex, which you refer to as a glycolipid, which can be on the surface of the cell. And these act as receptors on the cell for cell recognition. You do also have complexes of carbohydrates and proteins, which are glycoproteins. And so these two will be acting as receptors. So glycoproteins, glycolipids, they act as receptors for cell recognition. And you take a look at, you have some kinds of proteins here. You have a peripheral membrane protein, and you have, integral membrane protein. And you can see that for peripheral membrane protein, we'll come to look at these types of proteins even much more in detail. But you can see, at least you can just quickly notice that an integral membrane protein or transmembrane protein is embedded across the phospholipid bilayer, but the peripheral protein is only at one side of it. And then you can see another lipid, which we looked at. Cholesterol is also found in the biomembrane structure. So generally, these are the major components of the cell membrane, the biomembranes that you refer to. So you see, you can understand why, especially for those of you who when you go to do your project works and you do some extractions from plant leaves, things like that, and what you are extracting is some oil that later you want to analyze microbial activity of that. I mean, those of you who are interested in the microbiology aspect of your program and you go to do your projects in that field, you do some of those things where you test some extracts against some microbes. And a lot of these extracts that you are doing contain some lipids. And where are the lipids coming from? These lipids are coming from the cell membranes. So the membranes of the cell, or let's say the plant cells, that is where you are getting these lipids from, and you are testing its activity. Or maybe even if you go into cosmetology, for instance, that's there are some extracts from plants, some oil extracts from plants. I mean, where are these oils coming from? I mean, if it is extract from, say, the leaves, the flowers, they are coming from the cells, the cell membrane of these plants. Even if you take, say, maybe the seeds, okay, that one is storage. So you have these oils that are actually stored there in the tissues of those seeds, or maybe the pulp, the fruits, like maybe palm. And you have oil which is stored there. But if it is some essential oils that you get in very minute quantities, you should understand that these oils are coming from the cell membranes of the plants. That is where you see they are stored in very large amount. As you can see, phospholipids is a major part of it before you have some other components that are embedded in it. Now, with that composition of biomembranes, you want to understand that the composition of biomembranes is not a constant thing, that if you take every cell, the amount of some lipids, say the amount of lipids or whichever component proteins that you have in these lipids are going to be the same. No. You have different compositions. So it can depend on even the type of cell that you are looking at. That is, even within a particular organism, you have different cells that will have different compositions. So you have these, biomembrane compositions being different. Yeah, it's not constant. So, for instance, if you take an example of, the a rat hepatocyte, say a liver cell of a rat, which have been studied to see the composition of its cell membrane and also the membranes of organelles in that cell. You see that you have different lipids of different percentages that are in the various organelles. So for instance, if you take the Golgi apparatus, you can see, so you have the key below that you have cholesterol, just about 5-10% in there, and you have phosphatidylcholine, which is about some 30% in there, and you have phosphatidylethanolamine, also about 20%, and you have some minor lipids, about 10%, and you have some sphingolipids, also about 10%. And you can do the comparison for all of these organelles, say the inner mitochondrial membrane. So even if you take inner mitochondrial membrane and the outer mitochondrial membrane, it is the same organelle, but this organelle mitochondria is having two membranes, inner and outer membrane. And you see that the compositions are not the same. If you pay attention, you see, I send you the slide, you take a look at it and you realize that you have different compositions of even in one organelle, like the mitochondria, where you have the inner and the outer mitochondrial membrane. Similarly, if you take, for instance, the rough endoplasmic reticulum and the smooth endoplasmic reticulum, you see, I mean, it only means the rough endoplasmic reticulum is having some ribosomes on it, and you see that here too, the compositions are not the same. The same organelle, just some difference of attachment of another organelle there, and you see that there is difference in the composition of the lipids in the membranes there. Lysosome, lysosome, and the nucleus, all of them, they have different compositions. So, I mean, this is much something which you can quickly look at and appreciate the fact that the lipid compositions are not the same. But I would say that it's not a thing to be cramping in your head to say that you want to keep in your mind that if you take Golgi apparatus, it contains this percentage of lipids, and then if you take nucleus, no, you don't do that. You don't have to do that. You only have to just know that the compositions are not the same; they differ. I mean, that understanding is just okay. You can always refer to these things if anytime that you want to make some reference. You can just take a book, look into it, and see how much of lipids is here, how much of, and even specific lipids, how much of cholesterol do you find in a certain membrane. I mean, that is if you are doing some research and you want to do some extraction from some specific cells or specific tissues, then you just make a reference to see where can you get which specific lipid in large amounts, so you can focus on the extraction from that particular location. I mean, that is it. That is not a thing to cramp in your head. So that is to tell you, don't go wanting to say you want to remember the percentages of how much of lipids is in each of these organelles. You don't have to do that. All right. Another concept, another understanding you need to get is that if you take the biomembrane, you saw that the biomembrane is mainly a phospholipid bilayer in which you have other components embedded. Now, I mean, each of the layers is a monolayer, sometimes being referred to as a leaflet. Now, even if you take that, the cell membrane, which is of a lipid bilayer, both sides or the monolayers do not contain the same amount of lipids. So that is why you have what is being referred to as asymmetry. So the lipid bilayer, the cell membrane or the biomembrane is not symmetrical. you don't have the same composition in each of these leaflets or monolayers. And so here too, if you take an example of the red blood cells, for instance, and we're taking a look at the cell membrane of the red blood cells, and we are comparing the inner leaflet or the inner monolayer and the outer monolayer, looking at the different lipids that are in the cell membrane of the red blood cell, you realize that you have different compositions. If you take phosphatidyl ethanolamine, you can see that it is more in the inner layer than in the outer layer. I mean, if you can see maybe in the outer layer just about 10%, and you have in the inner layer about 90% there. But even all, if you take the percentage of the lipids, together in the membrane itself, you look at it, phosphatidyl ethanolamine is about just 30% in the cell membrane. So out of that 30%, you see that you have 90% of it being in the inner layer, whilst just 10% of it is in the outer monolayer. Similarly, if you take phosphatidylcholine, the composition in the cell membrane altogether is 27%. And out of that, you see that you have about 90% of it or maybe say 80% of it inside the outer monolayer. And you can do that for all of them. Sphingomyelin, too, most of it is in the outer layer. You have phosphatidylserine, most of it is in the inner layer. All the others, phosphatidyl inositol 4-phosphate, phosphatidyl inositol 4,5-biphosphate, phosphatidic acid, all of it, you see that you have most of them, the composition in the inner layer than in the outer layer. Now, if you go ahead and even take a look at cells from different organisms, and we take, say, the human, let's say, myelin sheath. You have seen myelin sheath in nerve cells. This time around, it's not just the lipids, but even looking at composition of the proteins as well. So you see that even the proteins have different compositions in there, different composition in different cells. If you compare, say, the liver cells of a mouse is having more protein in it than the myelin sheath of humans. The maize leaf is even containing more. Yeast is having more proteins in there. You have the highest proteins in the cell membrane of E. coli bacteria. So, and then if you take a look at all these other ones, we have seen them before. Phospholipids, we've studied phospholipids, so we know the structure, I think. And we also looked at structure of sterols, except that we only looked at the general structure of sterols. Today we can look at, say, maybe different sterols, which particularly you find in plants. And then you can see that you have different sterol types depending on the organism. So you realize that you have cholesterol in animal cells. You find cholesterol in animal cells. You don't find, if you take a cell membrane of plants, the sterol which is there is different. It's not cholesterol. It's different types of sterol. So we can look at that. So you have cholesterol in humans. You have cholesterol in mouse. But if you see in maize, yeast, paramecium, protozoa, you see that there are different types of sterols there. And we did look at these lipids too. these types of lipids, composition of monosaccharide or disaccharide in complex with lipids. So we did see like galactolipids, which we said were mostly in constituents of chlorophyll. In the pigment in plants, you find these galactolipids in there. And we did see all these other ones, plasmalogens, triacylglycerols. I mean, these are storage forms of lipids. So all these lipids, we did look at them when we were doing intermediary metabolism last semester. So we will take a much closer look at, say, sterols in plants later. So you can remind yourself of the structure of, cholesterol. I mean, all sterols have similar structure, having the steroid nucleus, where you have these 4 rings, we refer to as the steroid nucleus. You have, 3 6-sided rings fused, and then the last one, which is labeled D, is a 5-sided ring. And you have a side chain on that last 5-sided ring. And it is actually this side chain, this alkyl side chain, which is different for different sterols. So that is what distinguishes the different sterols that we have. But the steroid nucleus is constant. That's what you find in all the sterols. And you also want to remember that the sterol has a hydroxyl group, which is that hydrophilic part of the molecule. Just like if you take the fatty acids, you have the carboxylic acid group being that polar parts, hydrophilic, and you have the hydrocarbon part, as a hydrophobic tail. The same way here, you see that you have cholesterol that is having a polar head, the hydroxyl group, and carbon 3 of the ring A, which is hydrophilic. And of course, we did also look at the fact that they do act as some structural components of cell membranes, and at the same time they also play some role in cell signaling, cell transduction, in cell recognition. We'll also see that they play a role also in the regulation of the fluidity of cell membranes. I mean, you want to remember that when we looked at lipid metabolism, we did talk about the fact that the melting point of the lipids depends on the molecular mass of the lipid, the fatty acids, or to say the number of carbons. As the number of carbons increases, you have melting point also increasing, and vice versa. And similarly, if you take unsaturated fatty acids that have double bonds in them, one or more, the more double bonds you have in a fatty acid, the lesser the melting point, because those bonds are easier to break than the single bonds. And so more unsaturation results into lesser or a decrease in the melting point, which also will give you the idea about cholesterol. Looking at cholesterol, the structure of it, these fused rings together. This is a big molecule, which will be much more difficult to break down. And so the more of cholesterol you have in a cell membrane, the less fluid it will be. So that's what accounts for the amount of sterol or cholesterol which can be present in a particular cell, which is a sort of adaptation of the cell. Can be even a whole organism. That's even taking a look at a single cell organism that is adapting to a particular environment. It can do so by the synthesis of specific amounts of cholesterol in it to regulate the fluidity of the cell membrane. And we'll take a look at those things much more. Now you have the sterols. So we did say that you have cholesterol in animal cells, but in plant cells, you have different types of sterols. And these are what we refer to as phytosterols. And so this is for you to see the different types of the sterols. I mean, most common of them. Of course, this is not all of it. You'll find more other, phytosterols, but these are common phytosterols that you come across. The beta cytosterol, the campisterol, stigmasterol, the brassicasterol. If you look at them, they all have similar structure. They have the steroid nucleus, which is constant. And you see it is only the side chain, which is on the fifth ring, the 5-sided ring, which is different. If you look at, you pay attention to it, you realize that that's the only, thing which is different in these sterols. It's what distinguishes them. Right. Now we can take a look at what we were talking about, that the sterols, or say if we're looking specifically at cholesterol, but even in plants where you see these different phytosterols, these sterols, we just saw that they help modulate or regulate fluidity. I mean, say stability of the cell membrane. I would take a cell membrane, looking at the fact that you have these fatty acids, or say it's phospholipids, which are arranged such that you have, the polar heads on one side. But in this case, we are even looking at it not as it's looking more as just a fatty acid and not even a phospholipid. Of course, for a phospholipid, you have a glycerol backbone most of all, and then you have maybe three or two fatty acids attached on it. There's a phosphate group, and then you have on that phosphate group another lipid or a different biomembrane attached on that to give you the whole structure of a phospholipid. We did look at that last semester, so we don't have to go back to that. But you have to go and do that revision to understand that you have polar heads that are arranged on one side, and you have the hydrophobic tail on the other side. Now, if you have such an arrangement, and you look at the structure of the sterol, because we did see in the structure of the biomembrane this arrangement of the lipids, and then in between them you have some cholesterol somewhere, and you see the cholesterol is occupying such a big a big surface area between two fatty acids, or say phospholipids. And because you have those 4 rings, and you can see those 4 rings are occupying such a large space. And then you have a side chain also, which is occupying such a space. And with such hydrophobicity and the space that they occupy, they are able to regulate the fluidity of the membrane. The more of these you have, the more of these sterols you have in cell membranes, the lesser fluid the membrane is going to be, meaning the membrane is going to be more stable. But of course, that is in relation to the kind of environment, conditions in which the cell is, or the organism is growing. So we'll look at some example of that later. Now we have what we refer to as transition temperature. So you see, we did talk about the fact that you have the molecular mass or number of carbons in fatty acids influencing the melting point of these lipids. So you see, if you have a number of unsaturated fatty acids, so you see, if you take a look at how the membrane behaves at lower temperatures, the membrane will be going towards solidification, because of course at lower temperatures there will not be enough agitation. There will be less amount of energy for the movement of the membranes, because the membrane is fluid, it moves, and so if you have lesser amount of energy that is at a lower temperature, then it will be turning more solid, less fluid. Then at higher temperatures, you have the cell membrane being more and more fluid. And so you see the state of the membrane on the vertical axis, more solid, or let's say less fluid below. So you have a less fluid character and a more fluid character above, depending on temperature. You have the mid temperature there being referred to, what is here, Tm, is what is being referred to as the transition temperature. That is, at that midpoint, you have, you can look at it as the optimal temperature at which the cell membrane is at, say, a normal movement of the membrane components, the lipids, happening at that temperature. If the temperature goes a little higher above that, then the cell membrane becomes a little bit more fluid. And you can see it gets much fluid as the temperature increases the more. You remember we did talk about, say, denaturing of biomolecules like proteins with high temperature. So you see, this is another thing that can also happen. If you have a cell membrane, that is what is keeping the cell together. It's because the outer membrane that is separating the cell from its external environment. And you have high temperature. So such a high temperature can result into, a breakdown of the cell, like cell rupture. You can have that occurring. And if you have a less amount of temperature, you see the cell is also becoming less, the membrane is becoming less and less fluid, which is also going to result into a decrease in the transport of substances across the cell. Now, looking at this, if you take the role of sterols in this, what do sterols do in terms of stabilizing the cell membrane in this instance? Now look, the same thing, the same chart that we just saw, the same graph we just saw. In this case, if you have sterols that are in the cell membrane, what the sterols do is that they widen the temperature around that transition temperature. That is to say, they give a wider range of the transition temperature, so that because you have sterols that with a structure, with each structure, it requires a higher amount of energy in order for them to become fluid. And so even if temperature increases, the cell membrane will still be stable much more than if there was no sterol in it. Similarly, when temperature is lowered, when temperature decreases, the cell can still withstand lower temperatures, because you see, with a structure of sterols, you require a large amount of energy in order to either break it down, or in order for you to say you are causing any form of solidification or freezing to it. The energy range within which you can have a change in fluidity of the cholesterol, or say the sterol, if it's in plants, you have different sterols there, that range of temperature is wider. And so that is how you can have the sterols regulating the fluidity of the cell membrane, or the biomembrane. All right, now we take a look at factors affecting membrane fluidity, what we just talked about. If we bring all of it together, we can simply say you have these, 3 factors that affect membrane fluidity. Temperature, we talked about it. The higher the temperature, the more fluid. Even looking at those charts, the more fluid the membrane will be. If the temperature goes down, the fluidity of the membrane will decrease. If you take degree of saturation as the second factor, which is affecting membrane fluidity, we did talk about the fact that the more double bonds you have in fatty acids, or let's say the more unsaturated fatty acids you have, the more fluid the biomembrane is going to be, and vice versa. The less saturated fatty acids, the more saturated fatty acids you have, the less fluid that membrane is going to be. That is to say, if you have lesser amount of unsaturated fatty acids with double bonds, if you have less amount of them in a membrane, then you are going to have that membrane to be less fluid. And we did also just look at, the sterol component of the membranes. The more sterol you have in a membrane, the less fluid that membrane is going to be, because the sterols will require more amount of energy in order for that molecule to be agitated to become more fluid. And we'll see that experimentally. We'll see that experimentally. This some experimental values from a study on E. coli, the bacteria E. coli. So, how you do culturing in microbiology, culturing of these microbial cells. This has been a study on, E. coli cells, where E. coli cells had been cultured at different temperatures. And the composition of the cell membrane of the E. coli cells at these different temperatures were then studied. And you see that there are differences in the composition of these E. coli cells depending on the temperature under which they were cultured, which is to say the E. coli cells actually adapted. They got adapted to the environment, the temperature in which they were being cultured. So they synthesized more of some lipids than others, depending on the condition in which they were. But most importantly, what we want to focus on is this last one, the very last row, which says ratio of unsaturated to saturated fatty acids. That is the most important, thing to look at here. And you see, you have E. coli that was cultured at 10 degrees Celsius. You had 20 degrees Celsius, 30 degrees Celsius, and 40 degrees Celsius. Now let's come to the ratio of unsaturated to saturated fatty acid ratio. So it's a fraction. You take, say, amount of unsaturated fatty acids divided by amount of saturated fatty acids. I mean, the higher the value you have will mean that you have more unsaturated fatty acids. That is, more of fatty acids with double bonds in them. And what do you notice? You notice that as the temperature increases, you have lesser amount of unsaturated fatty acids synthesized by the bacteria in its cell membrane. And why so? So look at it. If you have 40 degrees Celsius, which is higher than 30 degrees Celsius, what it means is that that's a higher temperature. And at that high temperature, the biomembrane components are going to be more agitated, which means the temperature will cause more fluidity of the cell membrane. All right, so if the temperature will be causing more fluidity of the cell membrane, then the cell, the organism, must adapt to that higher energy. And so to adapt to that higher energy, the organism synthesizes more saturated fatty acids. And so that is why you have the value decrease. Because more saturated fatty acids means the denominator will be greater. And that will make that ratio to be less. And so you see, as you go along as the temperature increases, you see that ratio decreases. From 10 degrees, where you have 2.9 for that ratio of unsaturated to saturated fatty acids. That is high, meaning there were more unsaturated fatty acids. More unsaturated fatty acids synthesized by this organism because at this lesser temperature, there will be lesser agitation of the membrane components, which means there will be less fluidity. Now, if there's going to be less fluidity, then the organism must adapt by producing more unsaturated fatty acids that contain more double bonds that will not require higher amount of energy to be fluid. So you see, this is how an organism, say a bacteria, can also adapt, to such conditions. And even if you take the slide and you just want to, take a look at the composition of saturated fatty acids at the various temperatures, between the, for instance, if you take oleic acid, which is having 18 carbons and 1 double bond, that's an unsaturated fatty acid. You just want to see how the trend looks like. That's unsaturated fatty acid. At 10 degrees Celsius, you have more of it, and you see it keeps decreasing when the temperature increases. 20 degrees, it became 34%. At 30 degrees, it became 30%. And at 40 degrees, it reduces much, much more significantly to 12%. And you see the same thing for, say, palmitoleic acid, which is also an unsaturated fatty acid. You see the same trend, that the percentage keeps reducing as the temperature increases. And how about saturated fatty acids? You see saturated fatty acids, the opposite is happening. You see, as the temperature increases, you have the amount of saturated fatty acids also increasing to compensate for the increase in temperature. I mean to compensate for that fluidity. So you see, from 18% at 10 degrees Celsius, it increases to 25%. That is for palmitic acid. 16 carbons, no double bond. That's a saturated fatty acid. And the percentage increases more when the temperature was 30 degrees Celsius, and increases much higher to 48% when the temperature increases to 40 degrees. So, I mean, when you get a slice, you can pay attention to some of these trends and try to understand, listen to the lecture again. If you are not getting it so well, you have the opportunity, listen, take the slides, take books, read, and understand. Now, with what we just looked at, we can look at what we refer to as membrane dynamics, where we look at movement of the components of the membranes, particularly the lipid components of the membranes. And we see, we also looked at this transition temperature and the dependence of fluidity of the membrane with the temperature. And if you put all of these together, you can have biomembranes having two major states. You can have biomembranes having a gel or a semi-solid state. You can have them having what you refer to as a liquid ordered state, or you have them having what you say a liquid disordered state, which is the like say more fluid state. That's at higher temperatures. Now, this is shown at the right-hand side just to give you an idea of what is being talked about. Now, see, if it's a fluid state, the molecules are moving, so fast. So if you take an image of it, if you take a microscopic image of it, you really do not see the molecules that distinctly. That's a fluid state of the membrane in such high temperature. But if it's at a low temperature, where you have that, a gel or a semi-solid phase of the biomembrane, also, looking at it as a paracrystalline state, you can see that the molecules are looking more distinct. I mean, the phospholipids are looking more distinct there when you take such a microscopic shot of it. And so you see, you have heat or thermal energy, resulting into such changes in the phases or in the states of the membrane. Either changing from gel to fluid, or vice versa, depending on the temperature. You do have that occurring. So you can pay attention to the fact that you can have these states of biomembranes. The gel or the semi-solid state. You can have the liquid ordered state, which is intermediate with temperature. So let's say just about that transition temperature, the Tm, which we saw on that chart. You'll be having this. It's fluid. That's okay. It's ordered. But if the temperature goes high, that the biomembrane, lipids become more fluid. The movement is much more because these molecules are more agitated. And of course, you are going to have what is being referred to as a liquid disordered or a fluid state of the membranes. Now we can talk about the movement of lipids, the types of movements of lipids in the biomembrane. We can categorize the movement of lipids in biomembranes into two major groups. The first we refer to as uncatalyzed movements. And what it means is there's not going to be any participation of an enzyme in that process. Then you have catalyzed movements. So here you have the movement of the lipids, being catalyzed by enzymes. So you see, again, we are building on. That's why we studied some things on enzyme before. So now we are mentioning them again, that they have a role to play in some of the movements of the lipids in biomembranes. And we'll take a look at these different types of movements of lipids, what we refer to as a transbilayer diffusion, or it's also referred to as a flip-flop movement. You have this transbilayer of lipids, and so if you have a movement of lipid from one monolayer to the other, that is transbilayer, from one layer to the other. Or you have a lateral diffusion, where you have the movement of the lipid in one of the layers. It doesn't cross to the other. It's just moving along that membrane, that monolayer. That is a lateral diffusion. Then you have the catalyzed movements. You also want to pay attention to the fact that you have two of these catalyzed movements that are ATP dependent, meaning there is expense of energy in the form of ATP. And you have one of them which is ATP independent. So even though it is catalyzed, there is no use of energy in this form of movement, this catalyzed movement. We will see we have a flip movement, we have a flop movement, and then the ATP independent is flip-flop, which means it will be in both ways. And we'll get to understand what we mean by flip and what we mean by flop. And we're looking at the first uncatalyzed movement, the transbilayer diffusion, what we refer to as the flip-flop. Now, what do we mean when we say flip? Now, if you want to remember, just understand. You see, flip is having I as the vowel there. Flop is having O as the vowel there. So if you take a cell membrane, I means in and O means out, which is to say that flip refers to the movement of the phospholipid, a phospholipid, from an outer membrane, that is the outer monolayer, to the inner monolayer. That's what I said. Just use the letter, the vowel I. I means in. So from the outside monolayer to the inside monolayer, flip. But if the movement is in the opposite direction, where there is movement of the phospholipid from an inner monolayer to an outer monolayer, that's a movement out. That movement is what is referred to as flop movement. But the two of them can occur. That's why it's just being referred to as flip-flop. Flip-flop movement as an uncatalyzed movement. This is not a movement which is catalyzed by an enzyme. And you can see that movement is very slow. It can take days. It's uncatalyzed and it's spontaneous, but it is very slow over days that these can occur. All right. Now we have the lateral diffusion. So we take the cell like a sphere, where even though the arrow is showing just along the membrane, of course, a 2D version. I think when I come and we meet face to face, I'll show you some animations so that you can actually see much better. How, the cell is like a ball in which you have a nucleus and the organelles inside. And so if you are saying that movement of phospholipid bilayer with, say, lateral diffusion, that is in one monolayer, it's not just that because the arrow is to the right or is to the left, the movement is to right or left, but the movement can be left, right, front, back, because it's a sphere. So you have the movement of these lipids in different directions, but it is in one of the monolayers. It does not cross to the other. That's why it's lateral diffusion. It's not transbilayer. And this movement is fast. See, 1 micrometer per second. It's a much more faster movement than that of the flip-flop. Right. Now we can go to the catalyzed movements. The catalyzed movements. Now the first catalyzed movement we are seeing here is with flippase. The enzyme is flippase. And that enzyme catalyzes flip movement. And flip movement is what? From outside to the inside, in. So the enzyme there is flippase. Then you have floppase. That catalyzes the movement of a phospholipid from inside to the outside. So that's a flop movement. And remember that these two movements, the flip movement, the flop movement that are catalyzed by flippase and floppase, are ATP dependent. There is expense of energy in these movements. But when you come to the catalyzed ATP independent movement, that movement too can be in both ways. It can either be a flip movement or a flop movement. But this is catalyzed by a different enzyme, scramblase, which means scramblase can do what both flippase and floppase do. Scramblase can do that, and scramblase will do it without the expense of energy. So these are some 3 different enzymes that you are learning of today: flippase, floppase, scramblase. All right, so you should be connecting the things as we study them over the period. Like, last semester when we did, look at intermediary metabolism, we did some studies on enzymes, and you did see the naming of the enzymes, where you had ACE, the suffix ASE, added to either the activity of the enzyme or the substrate upon which the enzyme was acting. Enzyme naming, and you can see it here too. Flip movement, that's an activity, you say flippase. Flop movement is an activity, you say floppase. That's a scramblase, more like it's scrambling. It's either is in or out. It's not flip, it's not flop. It can be both. And that name is given to it as scramblase. All right. Now, we can also talk about what we refer to as hop diffusion. Since we're talking about movement of lipids in the biomembrane, we have what we refer to as hop diffusion, which refers to, rapid diffusion of lipids within some confined regions. So these two had been studied with some microscopy to see that actually, if you track the movement of a phospholipid in a cell membrane, you realize that the phospholipid is moving around a particular area for a long time and does not just move at very large distances. So you see it will be moving just within a particular area for a long time, and then occasionally it will hop to a different region for some time and then come back. And so you see this phospholipid, which, let's say, the yellow or the orange movement is looking at the movement of one phospholipid. The green is also looking at one movement of a phospholipid. The blue, the same, and then the violet. And you can see they are all moving within same regions for a very long time, and then they hop to one place and then they come back. So you see that at a particular place, there is really no space there. You can see they've been in that space for a very long time. They hop somewhere and then they come back to it. That's what is being referred to as hop diffusion. And with that, the term molecular fences was coined theoretically, to say that there is some form of a molecular fence, like a fence within which you have these lipids moving, so that they don't cross these fences. So you have a particular area within which a particular phospholipid moves. All right. So I think okay, we can quickly. I will just quickly refer you to go and do some revision, because I think when we did lipid metabolism, we did look at membrane lipids and some disease conditions that can occur with some abnormalities in the metabolism of some of those lipids. And to make it more practical, you see that we did look at this phospholipases, different ones, like phospholipase A, responsible for deesterification of the first fatty acid from the glycerol backbone. Phospholipase A2, responsible for, that's hydrolysis of, you can say hydrolysis, you can say deesterification. These are ester bonds, and so you can say deesterification. The process is also hydrolytic, so you can also say it's hydrolysis. And so you do that, phospholipase A1, phospholipase A2. These are enzymes involved in the breakdown of phospholipids, specific bonds that they break. You have the phospholipase C, breaking the phosphoester bond between the phosphate group and the glycerol backbone. And you have another phospholipase D, also breaking a phosphoester bond, but this is between the phosphate group and the head group, which is on that phospholipid, which can be any molecule there. We did look at that. It can be even an amino acid. It can be carbohydrate. It can be another lipid. And so you have these different enzymes that do this, breakdown of these phospholipids, because the phospholipids undergo some recycling. Constantly they are breaking down and being synthesized. The component of it is not just constant. Now, apart from these enzymes which break down those phospholipids, we do have some membrane lipids, which I think we did look at this, where you have these membrane lipids, which are in some nerve cells. The membrane lipids with these are sphingolipids, the backbone being ceramide. You have a sphingosine with some lipids and some oligosaccharides attached, and you find them, say, in the myelin sheath, in the membrane of neurons, the nerve cells. Now you have some enzymes like beta galactosidase, which is responsible for, say, this particular membrane lipid, which is so it's a glycolipid, which is having some oligosaccharide attached on the sphingosine. You have the keys. If you have blue, blue means it's a glucose residue. Yellow means galactose residue. The light blue there is, or the marine blue is 5-acetylneuramic acid. And then the plain white is N-acetylgalactosamine. So these are residues within the oligosaccharide, which is on a ceramide, or say having the sphingosine backbone. And you have these glycolipids, and are going recycling constantly, so that if you have the enzyme which is responsible for, say, one of these reactions, which is to cleave off the terminal galactose residue there, to convert this oligo, or say this glycolipid GM1 to GM2. If you have that enzyme beta-galactosidase being deficient, then you have a condition referred to as a generalized gangliosidosis. And you have a similar thing here. If you have this enzyme hexosaminidase A being deficient, then you have the condition referred to as Tay-Sachs disease. These are some neurodegenerative disorders, causing some intellectual disability, some mental disorders, which, I mean, the molecular basis of it is what we are looking at. We are not looking at, say, what are symptoms and things like we are doing biochemistry, and so we are just looking at the biochemical foundation, the biochemical basis, what causes such conditions. Because what happens is, once you have a deficiency of this, say beta-galactosidase, and it's not able to break down GM1 to GM2, it means there's going to be accumulation of GM1 in the system, and the accumulation of GM1 has such neurodegenerative disorders. Similarly, the deficiency of hexosaminidase A will cause accumulation of GM2 in the nerve cells, in the nervous system, and that also results into some effect on vision, some blindness, deafness, and you do have also the deficiency of, say, glucocerebrosidase, which is also responsible for, say, breaking down this glucosylceramide by cleaving off that glucose residue from ceramide to give you a ceramide, which will go into recycling of other membrane lipids. And that condition, where you have accumulation of this glucosylceramide, is what is referred to as the Gaucher's disease. You do have another one where you have deficiency of sphingomyelinase, responsible for the metabolism of sphingomyelin, breakdown of sphingomyelin. And that condition is referred to as the Niemann-Pick disease. All of these are nervous system conditions resulting into different symptoms, particularly neurodegenerative disorders and intellectual disabilities, mental health disorders. You do have those things happening. All right, so we have come to the end of the lecture today. I've been talking throughout, so at this time, I'll keep quiet and listen to you. If you have anything to say, you can write in the chat, or you can raise your hand. Then I will enable your microphone. You unmute yourself, you speak, and I'll respond. The floor is open.